Good morning, everybody, and welcome to our first Diagnostics for Varroa webinar. My name's Tamara Prentice, and I'm the Transition to Management Coordinator based within the New South Wales Department of Primary Industries, but working for the national program. So just going to do a really quick overview of what we've got in store for you today, and then I'll hand over to David Britton, who's really the star of the show today, sharing his key learnings from our initial Varroa response to hopefully make your lives a bit easier. Just a couple of housekeeping items. If you use the Q&A function for questions, we will answer in the Q&A response chat where we can, and we'll also do a live Q&A discussion at the end of the presentation as well. We just wanted to also promote the National T2M program weekly newsletter as well. If you're interested in signing up for weekly updates about where the program is up to, there's a QR code on the screen, and we'll share this presentation afterwards as well for anyone that might be interested. Lots of useful information in there. So I guess I just wanted to give an overview about why we were here today. So the transition to management plan includes a lot of activities. You may be aware that in each of your jurisdictions, officers are being employed to help beekeepers on the ground with surveillance and monitoring techniques. And we're also running more formal training courses across the country to raise the awareness of both Varroa and the potentially complicating viruses as well. And I guess deformed wings probably the most prevalent in people's minds. We expect that as people's awareness of these complications goes up, that you'll see a spike in requests for diagnostics where symptoms are observed. So unfortunately for you, we expect that your jobs will get busier as we start to get more information about the potential complications from Varroa out into that beekeeping community. So today's workshop has been put together to share the lessons that we've learnt in the early New South Wales response and hopefully provide some practical considerations for you and your teams as this mite enters your jurisdictions and you're being put through that same ringer. On the screen, I've just put some of the key activities underneath that activity 3.3.1.5. So the other things that we will be doing as part of this activity will be providing a budget for sample processing. So this has been provided as part of those Varroa development officers that I mentioned a second ago as part of their budget. And that's to try and encourage beekeepers to submit samples at no or minimal cost. This is being, mentioned, as I mentioned, through the Varroa development officers mentioned earlier. So hopefully they can encourage some of your beekeepers on the ground where they're seeing things like bees with deformed wings to send a sample in for processing. We're also doing a benchmarking of viruses nationally project as well. So the idea here is to benchmark the viruses before Varroa spreads so we can get a baseline for where things are up to at the moment. We're currently working on this and you may have already been spoken to by somebody, but the jurisdictions are meeting next week to run through the possible project. Look, I guess to note for that one is we're proposing to process the national samples in New South Wales at EMAI for consistency. While we process, as the process is sort of already established there, but your jurisdictions will have a say on that. So don't be surprised if they reach out to you. Look, I think that's about it from me. And I might hand over now to David Britton, who will lead us through some of his key learnings from the response. Take it over, David.
Good morning, everyone. I'm the diagnostic entomologist at Elizabeth MacArthur Agricultural Institute for New South Wales DPI. This is a relatively new part of our plant health diagnostic services. Prior to that, diagnostics were done out of Orange. So when I started in 2022, they were already 1 month into the Varroa response. And in fact, one of my early encounters with DPI staff were a bunch of people in Cairns who got off the plane and said, "Oh, we've just had Varroa detected in New South Wales." And I thought, oh, maybe in a month or so, it'll have all died down. Unfortunately, that was not the case. So I was launched into looking at something I'd managed to avoid my entire professional career up to date. So now I know too much about it. Before this morning, I thought I'd just have a quick look at some of the stats that were involved in providing diagnostics for the program. So I just might share that with you before I launch into the presentation. In 2023 alone, and just at EMAI, and I will point out that Orange also did a lot of diagnostics of sticky mats. We handled over 23,000 sticky mats for diagnostics. The worst week, or the best week, if you look at it that way, we did 3,222 mats, and that was with, I think, 20 microscopes working and people doing overtime and working on weekends. And all up, I think we counted over 114,000 mites off mats for the whole of the response. So hopefully none of you will have to share that experience, and it'll be a much more toned down event for you. But I thought I'd give you some scary stats to begin with. So I'll just fire up my presentation. Okay. All right, hopefully that's all good for you. Many people participated in providing diagnostics for this and for setting up the systems that we worked with. We had, so we've got quite a list of authors there of this. So the response ran from 2022 to 2024, and it was a response that was aimed at eradicating the mite from Australia. So I think the surveillance requirements that you'll have in your own jurisdictions will be quite different to what ours were, where we were trying to delimit and eradicate. And that's going to change the way that you do diagnostics. So it'll be different to what we did, but I guess I'm providing you a worst case scenario here. So today I'd like to cover off on the initial detection, what happened next for us, the sample types that we received, the workflow that we used at EMAI to handle the samples, some of the issues we had with managing diagnostics, and just a bit at the end about training staff to do diagnostics for Varroa. So the initial detection was in June 2022 from 2 mites off sticky mats and sentinel hives in the Port of Newcastle. They were identified by our arachnologist, Lauren, to Varroa, and then transported to EMAI on the same day for CO1 sequencing on site. And they were identified morphologically as Varroa destructor by Jürgen Otto at DAF Mascot. And that was confirmed on the 23rd of June. So it was a very quick process from initial detection to confirmation. So throughout the response, and particularly early on, we did a bit of molecular work. So the CO1 barcoding approach provides very straightforward species level ID for Varroa. So that was a fairly simple, relatively simple thing to get out of it, although we had to do it in a hurry. However, trying to do more fine-grained, more fine resolution of where the mites might have come from and trying to identify the pathways that might have taken was much more difficult, and we haven't successfully been able to do this. So we did attempt whole genome sequencing, but no clear answers were obtained. So some of the issues which arise with Varroa is that we had limited sample sizes from some of the apiaries and hives that we had. So if you're dealing with only 1 or 2 mites, it's difficult to get enough DNA to do whole genome sequencing, and that is followed by just the problem of getting good yields from mites. They're surprisingly difficult. Even though there are large mites, it's difficult to get good DNA yields. The biology of the organism is a problem as well. So they disperse by adult females. So an adult female will move from hive to hive using the honeybees. And once she's in that hive, she'll frequently mate she'll produce eggs, and then those eggs will develop into females and males. The males never leave the cell where they've been born, and there's sibling mating. So there's a lot of inbreeding happening most of the year. And finally, there was difficulty in controlling variability between sites, so the number of hives sampled and the number of mites between hives present in those hives. So that has made it very difficult to get any good answers there. So what happened next in the response? So we received bags of dead bees and hive frames from the Newcastle hives where the initial detections were made. Early diagnostics of brood comb were done to determine if the mite had established breeding populations, and they had. And initially, the diagnostic workload was split between sites. So Orange Ag Institute were doing the sticky mats with EMAI handling the rest of the samples. And we received all sorts of samples throughout the response, some very odd samples at times as well, where we really didn't know what we were supposed to do with them. So I've got the whole list here, but yeah, I think there was probably more than one entire hive sent in. The euthanasia process used petrol, so quite often we got bees which were reeking of petrol. Cloth like chucks and other absorbent material was sometimes put in hives to test for chemical residue, and then sometimes field workers left tools and PPE and so on in with the samples, mainly to ensure that they were securely disposed of. So as the response progressed, we initially focused on bee washers and the bags of washed bees like this one here and brood comb, but then it switched pretty much to sticky mats as the surveillance tool of choice based on sensitivity and ease of use and diagnostics. So they became very much the predominant sample. So I'll just go through these different samples that we received and sort of talk about some of the issues that arose with them and some of the benefits and I guess of using those methods. So bees and bee washers. So with this method, bees are washed in the field by surveillance staff. And the submissions that we received at EMAI were vials or jars of the wash, like in this bottom here photo here, and bags of the washed bees. They were done with ethanol or sometimes with metho if ethanol was hard to get. And sometimes they could be pretty substantial. So we're talking 8 to 10 kilograms worth of bees in bags and then whole crates full of ethanol in jars. So in the lab, if we were washing the bees ourselves, we were using these geological sieves and we'd place 1 or 2 cups of adult bees in the top sieve, which had with a 2 mil grid and with a 200 micron sieve underneath. So, and once you got the filtrate from the 200 micron sieve, we'd wash that into a Petri dish with 70% ethanol and look under the microscope for mites and other organisms. And the same process was used for washes that were submitted in jars. So we'd just put them through that 200 micron sieve. We pooled hives unless submissions indicated otherwise. So you'd often get multiple hives from a case. With bees and bee washers, most of the problems were around the size of some of these samples. And because they were big, it was difficult to fit them underneath fume hoods to manage the fumes from alcohols and petrol. So it was quite awkward initially, and you can see that there was a bit of a workaround attempted here. I think this is a rubbish bin purchased from Bunnings with so some fairly creative approaches to sieving large quantities of bees. We had a lot of problems with submission documentation and labels being damaged by leakage. I think it's probably worth enlarging on that briefly, is that there's a fair amount of insistence from NATA and other accreditation agencies that we use permanent markers when we're writing paperwork and so on of diagnostic submissions, and that's usually things like biros or permanent markers. Unfortunately, they are far from permanent when they come into ethanol, and the one thing that survives ethanol very reliably is pencil, which they do not approve of because in theory you can change pencil on documentation. So anyway, it's a bit of a tension that's difficult to resolve, but it's probably worth considering how you bag those samples and how you prevent the documentation from being lost. Oops, go back, come ahead too. So some of the decisions that you might want to take in regards to bee washing is whether or not you rewash bees that have already been washed in the field. We did have 1 or 2 instances where a site went from being negative for Varroa to positive after it was washed in the lab, but it was a rarity. And in view of the volume of samples we had to manage, we made the decision to not rewash bees in conjunction with the response managers. Whether or not you pool or not pool wash samples, so whether you keep information from individual hives separate or you treat the apiary as an entire sample. And perhaps investing in equipment to minimize manual handling, like this is a particular version that was devised by the Northern Australian Quarantine Strategy in Cairns for sieving bees. And that's a steamer on a shaking platform. And something I'll mention in all of these sample types is what you retain from samples and how much of it you retain, because we ran into storage issues very early on with walk-in freezers full of bees and bags of bees and honey and bits of hives and so on. Brood comb from hives, we had a lot of variability in the size of samples. We asked that samples be frozen prior to submission, and we asked that we had a section of brood comb that contains drone cells, which are preferred by Varroa, so you're more likely to spot them if they're there. One of the things about brood is that you can see all the different life stages of Varroa, so males and immatures are present, and it's incontrovertible proof of breeding populations in a hive. So the process is you just uncap in the lab we were uncapping drone cells with forceps. We remove the pupae and larvae from within and examine them. And we worked off around 100 cells per sample, which is basically following what the National Surveillance Guidelines recommend. And with empty cells, you can still see traces of mites being present because they leave a distinctive excreta on the cell wall. So the problems in the lab with this is that the samples take a long time to defrost, so you have to make a decision ahead whether you're going to handle brood comb on the day. It's messy and slow and very sticky, and there's an extra training requirements on recognising the early stages in males. So our recommendations around this is that you go for around about a 15 by 15 segment of comb, which is about the size of something that'll fit in a 2-litre ice cream container. Keep them frozen until you get them into the lab. Once they've defrosted, you can't refreeze them again because everything turns to mush. And just have a firm decision on the maximum number of cells you're wanting to uncap per sample. And again, that retaining samples, what do you keep? So finally, sticky mats. Well, for those of you who are not familiar with sticky mats, they're around about an A3 size gridded sticky mat that's placed in the bottom of a hive and protected from bees by a mesh. There's a miticide strip placed in the hive, and they're left in there ideally for around 2 to 3 days prior to being removed and submitting for diagnostics. So the idea of the mesh over the top is to stop the bees from eating the mat. It's only partially successful some of the times, and we received some samples of mat that were quite tiny by the time the bees had made an effort. Hang on, and early on in the response, the mesh that we were using was galvanized steel, which was later on replaced by nylon mesh. So once they're pulled out of the hive, they're covered with non-stick paper, and this is often the paper that's supplied with the mat from the manufacturers. So in the lab, we cut these into quarters or halves so they can fit underneath the stereo microscope, and we look at them at low magnification. And you can see why they have a grid of a certain size. This is approximately one field of view of that microscope. So, and the Varroa stand out quite clearly. They're not hard to spot if it's a nice clean mat like this. So there was a fair bit of variation in the mats. We had yellow backgrounds or white backgrounds. Most of them were printed with grids, but we did have quite a few which had no grids on them. And as I mentioned, we had the nylon and metal mesh options as well, which so they turned up in the lab with that mesh still in place. So problems in the lab with mats. Well, packaging was variable, and sometimes early on it was a little overzealous, and it took almost longer to get the mats out of the package than it did to actually look at them under the microscope. I can say that mats without the pre-printed grids on them are horrible to use because we were drawing on those grids with biro, and it was messy, or we were using transparent templates over the top, and it was just a whole level of extra work that we didn't need at the time. The metal mesh was a WHS issue in the lab, with people getting jabbed by the ends of it. So again, avoid that. And we had some problems with some submissions not being adequately frozen prior to getting into the lab. So we had live bees and potentially live mites on them. We didn't want people getting stung in the lab. And there's also that biosecurity issue of transferring a pest along with the mats. And then you had mats that are left in the hive for too long, like this sort of thing, where they're obviously going to be much, much more difficult to spot Varroa on when they're covered with that level of debris. And sometimes this was unavoidable because, access to apiaries was not always consistent. Sometimes it needed to get the apiarist in there or a property owner to unlock gates and stop dogs from biting the surveillance officers. So it's quite understandable that that happens. It's just something to try and avoid. And finally, disposal of mats. Because we were handling so many, waste disposal became an issue. They should be treated as biosecurity waste. So we had to make decisions about how we were disposing of them. So some of the decisions you might want to make in advance of using sticky mats is whether you retain that hive level identification or whether you just treat apiaries again as one case and site to identify positive or negative. And again, that same thing of retaining samples. Do you want to retain your positive mats, and how do you want to retain them? Do you want to keep them as mats, or do you want to pull the mites off them? We did some other diagnostics. So there was a concern that Varroa might have come in with some other friends. So we screened them for other bee pests and diseases. So in one of the ones we invested a fair bit of effort in was looking for tracheal mite, using molecular and bee dissection methods. Tropolapse, of course, is another concern, and viral diseases. And I'm not talking much about viral stuff, or not talking about any of the viral diagnostics today, because that's going to be the subject of another presentation further down the track by our virologists here. So one of the things I'd note about this is that we had some issues with using qPCR methods for detecting tracheal mite in samples, and this is a known issue. So you get false positives sometimes when you test bees. So this caused a fair bit of panic, of course, and we had to confirm using dissection that the mites were not present. So there are other species of Acarapis, which are external parasites on honeybees and which are present in Australia. And just occasionally, some of the sequences align with those for tracheal mite, and that causes that false positive. As I said, that's a known issue, and a lot of people have put a lot of effort to try and get around that, because the recommendation of dissecting 50 bees per sample is essentially taking out one person for a whole day to do that work. So going on to the workflow at EMAI. So at EMAI, we're lucky to have a sample reception area. So they they manage all of us incoming submissions of animal and plant samples of concern. And they did a huge amount of work during the response managing all these Varroa submissions coming in. So they arrive, they're logged into our laboratory information management system, which is a piece of software called Sample Manager by Thermo Fisher. Once they're logged in there, they're allocated a job number, and that's also replicated as barcode stickers. The samples are listed, tests assigned to them, and all the submission data on where they came from and who collected them and so on is entered and scanned and checked against what is actually submitted. So, and then the samples are sent to the relative lab to entomology or virology. Sometimes the subsampling was done in our side, and then these were passed on to virology. And then, so the lab side of things happens. So testing is completed and documented, and the diagnostic staff edit that job in Sample Manager, so they assign extra tests if necessary, edit sample details, and they do the results entry. As part of the workflow, we have to enter our results into other systems, and I'll talk a bit more about that later and problems with that. And finally, we have a laboratory services, like a customer services unit, which issues the reports and invoices, and they also did a huge amount of work. And it's often invisible to us diagnosticians who are sitting in the lab just how much extra work around the management of sampling and invoicing happens with this large-scale work. So all the reports are printed and archived and scanned. Positive samples are labelled and retained in storage, as I mentioned previously, and then we had our waste treatment process to get rid of what's left. So that's the ideal workflow. But there were substantial amount of issues, and these are not unusual for responses, and I think most of you have worked on responses will be familiar with some of them. And we always try and improve between the next response, but it's a slow process. So I'm in no way being critical of our surveillance staff or other people when I'm talking about these issues. It's just part of the problem, and you need to come up with ways of working around it. So some of the issues we had with the submissions themselves was a big one was lack of adequate information on what diagnostics are required. So you just got a sample that turns up and there's no indication on what people want you to do with it. And it was particularly problematic when we were receiving samples that we couldn't do here. I've already mentioned damage to submission paperwork from leaking ethanol. Mismatch between what is described in the submission versus the contents of the submission. And again, an easy problem to happen. And just the sheer volume that you get during your response, and just the physical space that you need to be able to work on handling the submissions, and just the number of submissions you get. So our sample reception people set up a space at the back of their work area, which was essentially a large open area with fold-out tables to manage all the mats coming in. Sample sent to the wrong place, so we did receive a few chemical residue testing samples, which were supposed to be going to an EPA lab in Sydney, so we don't do chemical residues here. Missing metadata was another problem, so you couldn't link it to a case or to a locality or with dates, a very common issue with most diagnostics. And of course, we had samples that sometimes turned up in very poor condition, so decomposed or defrosted and refrozen. Again, something that's quite familiar to anyone who's worked in a diagnostic lab for plant health and animal health. So a recommendation around submissions, just that surveillance staff are appropriately trained for submitting samples. No submissions are sent in without a diagnostic request. Pens and pencils on internal labels and vials, or some barcoding label system that's indelible and not going to wash off. We used QR systems, but we could have made better use of them, I feel. So barcoding of field samples. Double bagging submission paperwork, again, that happened later on. We got better at that. And submission staff stockpiling samples and then dumping on the lab in one lot was created a lot of workflow problems. So we did end up having all or nothing in some cases. So it'd be quiet for a week and then we'd have too much. So I know it's a hassle to send stuff in sometimes, but it's better to split it into smaller chunks. And surveillance staff should have a list of the labs that need to do the diagnostics and what tests they can do. The next issue was a biggie for us, was data management. And we ended up with double, triple, or even quadruple handling of results in many cases, particularly positive results. So first of all, we had our formal results entry and reporting through our LIMS, through the sample manager. Then New South Wales uses MAX, in common with a lot of the other states. So we're also entering those diagnostic results into MAX. It's not integrated with Sample Manager. Then we had just an internal tracking process that we required. So internal tracking is required because none of our samples, none of our databases, either SIMS, our LIMS or MAX, let you know where a sample is in the process of things. You can see that when we had some really high volumes, we had this quite creative system of sticky notes with where things were at and whether we'd finished them or not. And finally, the other thing that was required was email notifications to response staff in regards to urgent high priority cases and detections. I don't think that's avoidable and it's a necessary evil. So recommendations around data management: have an integrated system. We've been crying out for this for a long time. It's problematic because we've got two proprietary pieces of software which are not talking to each other. We're hoping that that's going to be fixed in the future. We're hoping that another thing that's highly desirable is to have key variables that are searchable in all your systems. In the case of MAX and Sample Manager, you cannot search on case numbers in Sample Manager. You cannot search on job numbers in MAX. So have it configured so all those critical variables can be located in any of your systems. Make sure that all the right people have access to the systems and are trained to use them. Obviously, there's sensitive data in some of these, so it needs to be the right people who have access. But we spent so much time searching for data where someone could have, if they had access and knew how to search in that database, they could have done it themselves. Investing in barcode and QR systems, I've already talked about that. Finally, it's very easy to let this drop off, but keeping diagnostic records on what you did with each sample is really useful when you have to go back and explain a result later on. And I know this sounds really obvious and it's something that we do all the time for diagnostics, but when you're rushed and you've got so much going on, it's easy to let it drop off. And similarly, standardized reporting frameworks and text, it's something that we normally do. But again, if you can stick to it, it makes life so much easier in the long run. We had no real way of tracking submissions for this response, and so we didn't know whether it was still in the back of someone's ute in the field, or if it was with a freight company, or if it was in sample reception, EMAI, waiting to get processed. We have no way of telling that, yet we have barcodes and so on in our samples. So my recommendation there is that you could look into integrating any one of the very numerous freight tracking systems that are available commercially and have that integrated with your barcoding system that you're already putting on submissions. I've mentioned retention of samples. It's an ongoing problem for us because we've got a few freezer fulls of these things. There's a requirement to meet the basic sample retention guidelines, which is 2 years, I think, for specimens from a response. Samples have to be stored to support compliance actions that might be in place for a response. You might want to document the incursion of a novel pest, so as research reasons that you might want to do this. Baseline information for future research, one that we're concerned about, of course, is development of insecticide resistance. So if you know what the resistance is like in the initial or if you have information on resistance genes in the initial specimens, you can use that for later on. But of course, it costs, and there's space issues for storing all this stuff. And you need to be able to make sure that it's adequate storage as well, so that they're appropriately preserved. So yeah, like I said, you need a lot of freezer space for this stuff, but if you plan ahead, you can reduce that by removing mites off mats and only keeping subsamples of bags of bees rather than entire bags. So invest in rationalizing things before they go in the freezer. Small vials rather than big jars and mats, and have some expectation up front with your compliance staff and others in regards to what they expect to have stored. I mentioned disposal was a bit of a drama. So with sticky mats was the main issue, because we had a lot of negative mats that we didn't need to keep. So we ended up splitting them into the paper component, which is the mat, and then the plastic component, which is the mesh and packaging. So, and we've been incinerating the paper component on site and autoclaving the plastic and putting that into normal waste. So I'll finish off talking about our training. We had 6 dedicated staff employed to support diagnostics for the work. So we had a technical officer to manage the reporting workload and to manage the staff who were doing the lab work. And then we've had 5 full-time, mostly full-time technical assistants working on it. And then we drew from other people on site and from casual staff. And all of those staff were trained using the same methodology and workflow developed at Orange. So Orange did all that initial work in getting this up and ready for us. So, and that training consisted of some Varroa in a vial, some bees, honeybees with mites glued on them in appropriate places. And not all of them, but some of them. And similarly, some mites on sticky traps. So the idea was that people would be able to get an idea of how big the Varroa mite was in the vial, and then recognize it in the context of something that most people are familiar with, which is a European honeybee, and then finally be able to see those same mats on the sticky mat. So that was the logic behind it. But as the response progressed, we found that was inadequate because we were getting so many people in who were not entomologists. The training was developed in mind with people who had some entomology background, and quite a few people who'd never used a stereo microscope. So a few of the recommendations that we would have for enlarging on that training program would be just basic instructions on how to set up and use a stereo microscope comfortably and ergonomically. Some more examples of sticky mats with non-target things on them. I've got an example of brawler fly here, and on here you can see a pollen mite, which some people confuse with Varroa early on, next to a Varroa. Some trickier sticky mats, so ones which have a lot of gunk on them or partially obscured Varroa like this one. And finally, some training on recognizing the immature stages and males of Varroa. So I think I've got to the end of my presentation here, and what I do need to do is just acknowledge just how many people put in an effort to support diagnostics for this, and particularly our specimen reception and customer service staff here at EMAI, all the staff at Orange who helped get us on board with doing the sticky mat diagnostics, but especially the people who were silly enough to work full-time on the response looking at sticky mats. And most of them are still working with the department, and I'm full credit for them for staying sane for the duration of that work. And I'm happy to hand over to some questions now from people.
Thanks very much for that, Dave. That was amazingly interesting. I worked for a very short period of time out in the response and sent you lots of stuff that might not have been appropriately cared for. So Anyway, now I have a far better appreciation of the difficulties that you faced during that process.
It's all in the past now, so.
Yeah. So there's a few questions in the chat, and the first one that I was going to ask is, who developed the washing process, and how was this process developed?
I actually don't know the answer to that. Maybe Lauren or Chris can provide that, but I suspect it was based on what is used in New Zealand.
Yeah.
But I'll let them chip in if they wish to.
Chris, did you want to chip in there?
Sorry, I just missed that question because I was jumping off a help desk call and I'm just come back in. Could you repeat the question, please?
Yeah, do you know who developed the washing process, presumably in the labs, and how it was developed?
So there's a number of references that could be provided for alcohol washing of bees as a standard method for quantifying Varroa mite infestations. So that's the basis of it. But then when you get to the lab, it's applying that same technique of using alcohol to remove mites from the body of bees. But in this case, you're doing it often to the entire colony of bees to remove all the mites. So there's a range of different aspects to it, but it is generally based on a recognized field test.
Thank you, Chris. I guess, David, you mentioned the rewash of the bee. So while we're talking about washing of bees, we might just jump to the next question. So why was a rewash of bees undertaken in the labs?
Basically, I think because we were feeling our way in regards to what was required for diagnostics. So there wasn't anything written out for us to follow, and we weren't necessarily fully confident that field washing was removing all the mites. And certainly there's been a bit of work on how much washing you need to do to get things out in some of those larger samples where there were large volumes of bees. I think there was not necessarily a lot of confidence that they'd been washed adequately to dislodge all the mites.
Okay, thank you. Sorry that you're copying all of these questions, but how did you handle the sticky mats in the lab, and what would you change?
I think we had it pretty much sorted out in the lab. I guess the only thing I might sort of look at changing would be to was more around just whether or not, I think it's more a decision that needs to be made by the surveillance group as to whether or not you treat hives separately or individually. And I guess that depends on what questions you're asking. But I think we had it pretty much sorted out in the lab process. And most of the issues were around data management rather than the physical management of the diagnostics.
So I think you mentioned in the presentation that you would cut parts of the mat and...
Oh, yeah. Yeah, well, I'm not sure how you quite get around that. And it was individual preference whether you went for quarters or halves. Yeah, you could potentially, I guess, buy microscopes which have an arm system so you can put a whole mat under all at once, but I'm not convinced that it really saves you that much in the end. You'd need a lot more bench space to do that as well.
Yeah, okay. Thank you. There's a heap of questions coming in now, so one of the next, I'll just run down the list. Someone's just said, would it be a good idea to increase the awareness of the videos and other field staff so samples submitted in the future are more suited to lab requirements?
Yeah, sure.
I thought that might be the answer. Yeah, rightio. Well, we might have a think about doing that then. The next one, I think possibly is one for me to answer. What are the projections for the coming spring to the current situation and further spreading, and what response arrangements are in place if it's going to be a bigger issue? So We don't really know what's going to happen in the coming spring. We do expect that we will see spread. There's a lot of bees moving at the moment to canola and then down to almonds, or similarly back to canola afterwards. So we do expect we will see spread from that. We ran a webinar on Tuesday night, which talked about every single each of the Eastern Seaboard states' response arrangements. And so I'd suggest, if you wanted more information about that, that you could potentially rewatch that webinar, and I'll get someone to share a link for that in the questions and answer spreadsheet as well. So that hopefully might tell you what each state is proposing to do in terms of response. So hopefully, David, that's given you a chance to catch your breath. But should specimens be vouchered with collections, and how should this be done?
We have vouchered specimens with the collection at Orange, and I would expect that, being mites, that preserved in ethanol is the way to go. And certainly we've retained quite a few in freezers in 100% ethanol as well, so not just routine 70%, 80%. So, but yeah, we've vouchered a lot of specimens already, and we still, as you would have seen with the freezers, we still have a lot more to deal with. There are some sort of projects that are loosely planned for those, but nothing concrete at this stage.
Okie dokie. Why did field staff submit steel mesh to the labs? Can they be washed in field to collect the contents and recycled for use on replacement sticky mats?
Yeah, well, yeah, I can tell you that we all hated the steel mesh, and it was just, I think it was just, again, it was just what was available early on in a hurry. So the same as using mats without lines ruled on them, we wouldn't ever have chosen that if that was an option, but we just had to deal with them. So once the nylon mesh became available and everyone seemed happy that it worked, we never saw the metal stuff again. Yeah, there was discussion about recycling it as well, because there were quite a few boxes generated. Mostly, by the time we were receiving them at EMAI, it was all nylon mesh.
Yeah, okay. And I guess just for anyone that's on the call, we do have a stockpile of nylon mesh here in New South Wales. So if you're talking to your jurisdictions and they're proposing metal mesh, you're welcome to reach out and we can send you across some nylon mesh. We have a lot left over. So then the next question is, I'm an entomologist from the NT, and I would like to know about diagnostic and other operational training programs available. Who should we contact?
This is kind of it. No,
yeah.
I haven't actually done this program myself, but CSIRO have run a couple of bee mite identification workshops. So this would have been, I think the last time they ran them was in 2020 or so. It was a fair while ago.
Yeah.
But they have the expertise to cover off on the other pests that we haven't got yet. So Tropilolaps and tracheal mites. So and I'm pretty sure a few people on this meeting today will have done that workshop.
Excellent. Thank you. And look, if you've got anything specifically that you want more information about, for example, maybe our lab processes or the like, feel free to send me an email and I will chuck my email in the chat. We'll share that around, because we are looking to make sure we can share as much information as possible to make things easier for your labs when this spreads. So the next, there's quite a few here, Dave, but we will pull up at 12. On time. So can you just tell us a bit more about what the males look like?
Laura, can you describe that for me, please? I'm not sure she's heard me yet. She's still on mute.
No.
Oh, sorry.
I'm throwing you in the deep end there. Can you describe a male mite?
Yes, sorry. I just I typed an answer for that just before. I'm so sorry. Yes, so males are typically a bit smaller than the females, and their distinctive because the females are distinctive. So we know the female Varroa mite to be quite an oblong, almost crab shape, with the legs all sort of coming from one margin on the side, which is quite unique in the mite world. Whereas the males are more of a circular body shape, and they're often not as sclerotized. There's quite a lot of the adult females will be sort of like a dark reddish brown color, and also the males tend to have longer legs compared to their bodies, as opposed to the females as well. So once there's plenty of resources online to look up what they do look like, and once you get your eye in, it's pretty easy to see the difference between the two.
Thank you very much, Lauren, much appreciated. So then, David, going back to washing, with the washing method of the bees with ethanol, was there any shaking involved? And if there was there a pattern or a time to be followed regarding this?
We actually, for the ones where we were using the geological sieves, the endicott type sieves, we were just using tap water to run over the top of them. So it wasn't so much shaking them as washing over the top with tap water to dislodge mites, because the bees had already been in ethanol by the time we received them. We were pretty comfortable that everything was dead there. It was just a matter of physically loosening them. So we didn't use a shaker per se for those samples. But yeah, if you have a look at some of the available methods online, they have actually used quite vigorous shaking and over quite a lengthy period of time. So that automated method that I mentioned above, I think they were running that for 15 minutes per sample, which was a lot longer than what we were taking with just washing them on the sieve.
Yeah. Okay. Thank you for that one. There's just such a long list of questions here. Considering the workload with the sticky mats, would just washing of the bees be enough to indicate presence of the mite in the apiary, and then use other methods to target that apiary in detail?
I didn't make any decisions around surveillance protocols, but my understanding of it is that the mats are the most sensitive in terms of detecting mites.
Yeah. Excellent. What impact did the response have on BAU non-Varroa diagnostics?
Terrible.
Yeah.
To give you a perspective, this is my first winter doing diagnostics for DPI where we haven't had the response, and I'm just astounded at how much extra time I have. It's not a bad thing. It's very good in finally catching up on all the things I said I would do. So
yeah,
it pretty much ate into a lot of what we were trying to do outside of Varroa, and if we wanted to develop protocols and training for other biosecurity pests of concern, it was almost impossible with that workload.
Yeah. No, it was massive. Look, we have got another 17 questions in the chat, but in the interests of time, we might pull up there, and what we'll propose to do is we'll send responses to each of the questions that we haven't got to, along with a link to the copy of this presentation, and also a link to that webinar I mentioned earlier about the response arrangements with the other states, to everybody who's attended today. So that way you'll all have it on record for future use. I just wanted to thank everyone for coming along to today's webinar. It's been great. If you wanted to reach out to me, and we'll send my details in that email as well, to let me know if there was anything specifically more that you wanted from us. We're more than happy to try and make sure that we can bring you guys up to speed as much as possible before you go through the same saga we have. So look, thank you very much to David for doing this presentation today. The feedback so far in the chat has been amazing. So I really appreciate your time, and thank you to everybody for coming along and listening in, and have a great day.
Thanks.